You may proceed.
Thank you very much, Your Honor. Good morning.
Good morning.
Could you please state your name for the record and spell your last name?
Bryce Raymond, Raymond.
Could you please tell...
Bryce, Bryce.
Could you please tell us your educational background?
I have a Bachelor's of Science in Forensic Science from Penn State University and a Master's of Science in Biology from Tufts University.
And what is your occupation, sir?
I'm currently a Forensic Scientist 2 in the DNA unit at the Massachusetts State Police Crime Lab.
And how long have you been employed at the Massachusetts State Police Crime Lab?
Since June of 2019.
And can you please tell the jury your present duties in the DNA unit and the nature of the work that you do?
Yes, as a Forensic Scientist 2, I am in charge with processing evidentiary items through our DNA lab processing, as well as interpreting and making any conclusions to known standards.
Do you have any specialized training to be a DNA analyst?
Yes, I received at least 6 months of specialized training in which I read all the protocols and procedures at the lab. I attended lectures and took quizzes, as well as worked on mock evidentiary items through the lab processing. And at the end of testing, I take a competency test.
And did you pass that competency test?
Yes, I did.
So does your testing and training stop after that initial 6-month period?
No, every year we have to do at least 8 hours of continuing education in order to stay knowledgeable in the field.
And do you continue to be tested at the lab?
Yes, we do.
And what kind of testing is that called?
That's called a proficiency test.
And how does proficiency testing work at the lab?
Yeah, so twice a year we have to take a proficiency test to continuously show that we have the skills and knowledge in order to perform DNA analysis. This is given by an outside accrediting body, and when we complete our work, we send in the results to that accrediting body for them to grade.
And what are the results of your proficiency testing?
I have passed every one I have taken.
So approximately how many samples have you had the opportunity to analyze for DNA testing?
Approximately 2,000.
And have you testified before in Massachusetts about DNA?
Yes, I have.
Does the Massachusetts DNA unit at your lab follow standardized protocols and procedures?
Yes, we do.
And who sets those protocols and procedures?
They are based on some accrediting bodies, such as the FBI, as well as validations that are done at the lab.
Well, you anticipated my next question, which is your lab, the Massachusetts State Police Crime Lab, accredited?
Yes, we are. We are accredited by ANAB, which is the ANSI National Accreditation Board, as well as the FBI Quality Assurance Standards for DNA laboratories.
And what does accredited actually mean?
That means we are following a specific set of standards to make sure that the scientific work that we are performing is accurate and reliable.
And is that a national accreditation?
Yes, it is.
And does your work undergo any kind of review process?
Yes, all work goes under 2 different types of review processes, a technical review and an administrative review.
And is that review process required under the accreditation standard?
Yes, it is.
And so if you could just explain technical and administrative review, please.
Yes, a technical review is one that is done by a qualified analyst that takes a look at your work to make sure that you followed all the proper protocols and procedures and made sure that any conclusions you made are scientifically sound. An administrative review looks over the file to make sure there's no spelling mistakes, no grammar mistakes, all the pages are numbered and initialed, and other administrative things such as that.
And why is that all important?
That's important to make sure that we provide the best quality work that we can.
I now want to talk about DNA. What is DNA?
DNA stands for deoxyribonucleic acid. It's the genetic blueprint of life and what makes you, you. You inherit your DNA from your parents, so you get half of your DNA from your mom and half of your DNA from your dad. And no 2 individuals have the same DNA profile except for identical twins.
Is DNA the same in every cell of a person's body?
Yes, so whether we test blood, saliva, hair, skin, we should expect the same DNA profile if they came from the same person.
So what type of biological material can DNA be found in?
So that can be blood, saliva, skin, hair, sweat, urine, anything biological.
So can you explain generally the process that is used for forensic DNA analysis?
Yes, at the lab we have a 4-step process. The first step is extraction. This is when we break open the cells and collect the DNA. The second step is quantitation or quantification. This is when we can get an estimate of how much human DNA is present as well as how much male DNA is present. And that's so we can target an appropriate amount for the third step. And that third step is amplification. This is when we make millions and millions of copies of specific areas on the DNA and are fluorescently tagged. And then the fourth step is separation and detection, also known as capillary electrophoresis. This is when we put the DNA on an instrument that separates out the DNA by size and has a camera to detect those fluorescently tags, and that produces raw data.
And what is the goal of that raw data?
The goal of that raw data is to then be read by an interpreting analyst in order to interpret the DNA profile.
And what is a DNA profile?
A DNA profile, so the specific areas that we are copying during amplification is called STRs, or short tandem repeats. That is just areas on the DNA that repeat different lengths for a person. So one person could have 5 repeats, another person could have 6. And so the combination of all those numbers is what makes up a DNA profile.
So how many locations are you testing?
We test 26 locations as well as a sex determining location.
And what is batch testing?
Batch testing is what we do at the lab and is a common practice in forensic science, in which if many items need to be processed for the same lab step, one analyst will process all those items at once on the same instrument.
So why do you use batch testing?
We do that for efficiency, as it is better for one analyst to process, say, 50 items at one time on one instrument than 50 analysts trying to process one item each on the same instrument, one after each other.
Is there a protocol that you follow prior to retrieving any evidence to prepare your lab area to avoid contamination?
Yes.
So how do you avoid contamination?
To avoid contamination, I first put on protective personal equipment, and that can include a lab coat, a mask, a hairnet, and for me, a beard net. When I go to my lab bench, I also put on gloves, and I spray the lab bench with bleach, clean that off, and lay down lab bench paper before I proceed with any testing.
So why do you use bleach?
The bleach is to destroy any DNA that may be present on the lab bench.
And why do you use bench paper?
We use bench paper to prevent any contamination from items that I may be testing to be deposited onto the lab bench for the next person.
And why are you wearing PPE?
The PPE is also to prevent any DNA that I may touch to go over to another lab bench or any of my DNA to be deposited on the items.
And once your area is prepared, can you please tell us what you first did and what you first do with items that you're going to sample?
Yes, so for sample preparation, I get a list of items that have been selected for DNA analysis. I get those items out of storage in our secured walk-in room. I take that container of evidence back to the lab bench. I scan it into my custody and take a photocopy. I then open up the container, grab the items that I need, and put those aside ready for testing. I seal up the container with my initials and date and put the container away.
And did you work on lab case 23-00596?
Yes, I did.
And what did you initially do to prepare the samples for DNA analysis?
I did sample preparations on some items.
Okay, so I'm going to ask you specifically about samples. Did you prepare samples 5-1.1, sample from swabs of red-brown stained cave on basement floor, 7-10.3.1, sample from the stained area C on the blade of the hacksaw, 7-10.4.1, sample from the handle of the hacksaw, 9-22.1.1, sample from stain A on white towel number 1 with red-brown stains. 9-28.1.1, sample from stained area A on apparent hairs. 9-31.1, sample from cutting of red-brown stain from piece of rug from bag number 5 from 300 Forest Street. Did you prepare samples from those?
Yes, I did.
So after preparing the samples, what did you next do?
After preparing the samples, I then did the first lab step of extraction.
And how did you do the extraction step?
Yes, to each of the tubes, along with the controls, I added chemicals to the tubes and put them in an oven, and that is done to break open the cells in order to free up the DNA. I then take those tubes and put them on an instrument that separates out the DNA and puts them in a separate tube in liquid form, and that tube is called an extract.
Did you use equipment?
Yes, I did.
What equipment did you use?
I used an oven, and the instrument was an Easy One robot.
And how did you use the equipment?
For the oven, I just placed the samples in and took them out. For the Easy One robot, I put the samples on the robot and then programmed the robot with the proper protocol to use for the extraction and collection, and then got the tubes off of the robot when the process was done.
And I think you said that that is now called, what is that now called?
That is now called an extract.
Okay. And then what did you do next?
Next, I took a small portion of the extract, known as an aliquot, and put that into a separate tube and prepared that for the second step of quantification.
So now we're on step number 2?
Yes.
Okay. And what items did you do quantification for?
The same items that were previously mentioned.
And how did you perform the quantification step?
Yes, so for each of the aliquots, I put a portion onto a 96 well plate along with other chemicals. I then placed that 96 well plate onto an instrument, and that instrument does a series of heating and cooling, and that produces the data that gives us the estimation of DNA.
So I believe you just named the equipment that you used.
Yes, it is a real-time PCR system.
Okay. And so how did you use that equipment?
So I put the 96 well plate onto the instrument. I then used the computer program associated with it to say that to start the quantification process. And then after it was done, I collected the data off of the equipment and then put it on our computer drive.
And then what did you do next?
After that, I discussed the quantitation results with the interpreting analyst of this case, who is summoned Salim.
And then what did you do?
Then we came up with how to do amplification strategies for each of the items. And then once that was decided, I went forward with the third step of amplification.
So step number 3 is amplification.
Correct.
And what items did you perform amplification for?
Those same 6 items.
That we previously named.
Correct.
Okay. And how did you perform the amplification step?
Yes. For each of those items, I got the extracts, and I either used all of the extract or a portion of the extract to target an appropriate amount of DNA for testing.
Let me just stop you for a second. What's extract?
Extract was the product from the first step of extraction.
Okay. And now finish your answer. I'm sorry to interrupt you.
Yes. So for those samples, I added chemicals and put them into an instrument known as a thermocycler. And that also does a series of heating and cooling, which makes the copies of the DNA that are fluorescently tagged. And at the end of that process, the tube is now known as an amplicon.
And then what did you do next?
Next, after the amplicons were done, I then proceeded on to the fourth step of separation and detection.
And which items did you do the fourth step for?
The same 6 items that were mentioned.
So you did all the steps of DNA for those 6 items.
That's correct.
And how did you perform the detection step?
Yes. Of the amplicons, I took a small portion of each amplicon and put it on a 96 well plate, added chemicals to the plate. I then put that plate onto an instrument known as a 3500 genetic analyzer. I then programmed the genetic analyzer to perform the separation and detection. And then the instrument separated out the DNA fragments by size and with the camera detected those fluorescent tags.
And then what happens next?
After that data is generated, I take the data off of the instrument and put it on our computer drive.
And then what happens?
After I'm done, I let the interpreting analyst know that data has been collected and I hand over any worksheets that I generated to them.
And who does the interpretation?
In this case, it was Summon Salim.
So you did not do the interpretation here?
I did not.
If I might have one moment, Your Honor. Nothing further, Your Honor.
Cross-examination.
Just a few questions. Mr. Rigg. Good morning.
Good morning.
And you've taken a lot of proficiency tests. You've passed them all, correct?
Yes.
You take pride in what you do, don't you?
Yes.
And you're running these machines, the analyzer and everything else, and you're running them consistent with the protocols that are required to be followed to run them accurately, correct?
That is correct.
And what you do is you depend on the fact that the samples that you get for testing are samples that will result in reliable results, correct?
Correct.
But what is your title at the lab?
I am a forensic scientist too.
Okay. And are there forensic scientists 3 and stuff like that? Forensic scientists 1?
Yes.
Okay, but you're a 2, correct?
Correct.
And with your knowledge and experience, it's fair to say, isn't it, that you have no idea where the samples that you subjected to DNA testing came from, correct?
That's correct.
And in fact, you have no idea how they were collected, wherever they were collected from, correct?
That is correct.
And in fact, you can tell the jury that at times when you conduct testing like you just described, the results will indicate that there are mixtures of different DNA in a particular sample, correct?
That is correct.
And whenever you get the results that result in a mixture, you can't determine, as a forensic scientist 2, or for that matter, forensic scientist 3, you can't tell how those 2 different substances came into contact with each other, resulting in a mixture, correct?
Our testing does not determine how DNA got onto an item.
And hopefully it's a simple question. Your DNA results do not tell you anything about what happened at let's just call it a crime scene, what happened at a particular scene or where they were collected, correct?
Correct.
Thank you, sir.
Any redirect?
No, Your Honor.
The witness may step down.
Thank you.
Good morning. Can you please state your name for the court and spell your first and last name?
Sure. My name is Brianna Kiesel, Brianna. Last name Kiesel, Kiesel.
And what is your educational background, ma'am?
I have a bachelor's in biology from Arcadia University and a master's in forensic science from Towson University.
And what is your occupation?
Currently, I'm a forensic scientist 2 with the Massachusetts State Police Crime Lab,
And what unit are you assigned to?
the DNA unit.
And how long have you been employed at the Massachusetts State Police Crime Lab?
6 years.
And what are your responsibilities as a forensic scientist too?
I am authorized in processing, interpreting, and reporting samples that were collected from crime scenes, as well as standards collected directly from an individual. I can also technically review other analysts' work when it concerns batch files, which we call files that have standards collected directly from an individual.
And do you have training for this position?
Yes, I do.
Can you tell me about your training?
Sure. Our training is a minimum of 6 months, and during that time we have lectures, quizzes, and this all culminates into a final written exam. We also have a practical component in the laboratory where we take several steps for sample sets forward, and that all culminates into a final written exam, as I mentioned, and a final lab practical.
And did you pass that written exam?
Yes, I did.
And that's called a competency evaluation?
Yes, that is a competency, the lab portion of it.
And do you also have to take proficiency testing?
Yes, we do.
And can you explain that to the jury?
A proficiency test is an ongoing competency test where we have to take samples forward twice a year to prove that we are that we are knowledgeable and still performing that skill or method according to the laboratory's standard operating procedures.
And have you passed your proficiency testing?
I have, yes.
And have you testified in court about DNA?
I have.
So I want to now talk to you about this case, 23-00596. Prior to retrieving any evidence in 23-00596, how did you prepare to process the evidence?
So once I was assigned the samples, I cleaned my bench space with bleach and wiped everything down, every instrument that I was using. And once that was done, I laid down a piece of fresh bench paper.
And directing you now to lab number 23-00596, did you work on items 9-27.2.1, the sample from the interior of the dark gray slippers from bag 4 of 300 Forest Street, and item 9-27.3.1, stain A of the exterior left gray slipper?
Yes, I did process those samples.
And what work did you do on those two items for sample prep?
For sample prep, they were already prepared by our criminalistics unit, so I just went into a secure freezer area to retrieve the packaging. I removed the tubes from the packaging and just visually examined the items within the tube.
Did you also work on item 9-32.1.1, the sample from the interior sleeve cuff of the Tyvek suit, 9-32.2.1, the sample from stain A on the exterior left pant leg of the Tyvek suit, and sample from stain D on the interior right sleeve of the Tyvek suit?
I did, yes.
And what work did you do for that?
Those samples I performed just like I previously mentioned, where I went to the secure freezer area, retrieved the packaging, removed the tubes from the packaging, and just observed the samples within the tube.
So you also just did the sample prep for that?
Yes.
Did you also work on item 16-13.1, the unknown tissue recovered from the Swampscott dumpster?
Yes, I did.
And what work did you do on this item?
So in this case, this item was still in a specimen jar, so I retrieved the packaging just like I previously mentioned, made sure the seal was secure as I did with the other samples. And then after that, I opened up the jar and sampled a few items from the jar and placed it into a tube for processing.
So you actually had to make the sample for this?
I did, yes.
Okay. And how many samples did you make?
So this was 16-13.1. There were two tubes we referred to as 16-13.1A and 16-13.1B.
And did you do any further work?
No, that is where my work ended.
Okay. And where did the samples then go?
I then passed the samples off to Madison Frank for extraction.
Cross-exam.
No question.
The witness may step down.
Thank you.
Can you please state your name for the court, spelling your name?
My name is Madison Frank. That is MADISON FRANK.
And what is your educational background, ma'am?
I have a Bachelor's of Science degree in Forensic and Investigative Sciences from West Virginia University.
And what is your occupation?
I'm a forensic scientist too in the DNA unit of the Massachusetts State Police Crime Lab.
And how long have you been employed at the Massachusetts State Police Crime Lab?
I've been employed there since October of 2019.
And what are your responsibilities?
As a forensic scientist too, I will perform DNA analysis on either known or evidentiary biological samples, interpret those results, and report out my results in a case file.
And have you had training for this position?
Yes, I have.
And have you passed your proficiency testing?
Yes, I have.
And have you passed the competency testing that was part of your training?
Yes, I have.
And have you testified in court about DNA?
Yes, I have.
Directing your attention to lab number 23-00596, did you do work on this case?
Yes, I did.
I want to ask you now about items 9-27.2.1, the sample from the interior of the dark gray slippers from bag 4 of 300 Forest Street, 9-27.3.1, the stain A on the exterior left gray slipper, 9-32.1.1, the sample from the interior sleeve cuff of the tie-back suit, 9-32.2.1, the sample from stain A on the exterior left pant leg of the tie-back suit, 9-32.5.1, sample from stain D on the interior right sleeve of the tie-back suit, and 16.13.1, the unknown tissue recovered from the Swampscott dumpster. I believe I just gave you 6 of those samples. Did I mention 9-32.2.1, the sample from stain A on the exterior leg pant of the Tyvek suit? So those would be the 6. What work did you do on those?
DNA analysis is a 4-step process, so I performed steps 1 and 3 of that process on those samples.
Okay. And what are 1 and 3?
Step 1 is extraction, and step 3 is amplification.
Okay. And how did you do step 1, the extraction step?
For extraction, I received the samples and I added chemicals to the samples and the controls, and then put them in an oven so that we can break open the cells to obtain the DNA. Then those samples will go on an instrument that helps to clean up the sample so that we're only dealing with the DNA and no other cell parts. Then after the extracts come off of the instrument, then I will take a small portion of that sample and I will put it in a new tube to be used for the second step.
And which instrument did you use or equipment?
An Easy One robotic instrument.
Okay. And how do you use that instrument?
The samples will go onto the instrument in tubes, and then there will be new tubes where the samples will go through processing in the instrument to put out just the liquid and DNA portions of the sample.
And then what is that biological material then referred to?
An extract.
And what is that extract then ready for?
The extract is then ready to be quantified in the second step.
So did you do that second step quantification?
No, I did not.
Who did that second step for those 6 items?
Troy Adams did the second step of quantification.
And why is that?
We use a batching process when we are processing our samples so that it is a more efficient and streamlined processing.
And are you qualified to do all of the steps of DNA, the 4 steps that you described?
Yes, I am.
Okay. And then did you do more work on this case with the 6 samples?
Yes, I also did the third step of amplification.
On all 6 of those samples?
Yes.
And that being 9-27.2.1, 9-27.3.1, 9-32.1.1, 9-32.2.1, 9-32.5.1, and 16-13.1. Did I get that right?
Yes, that is correct.
Okay. So you did the third step, and how did you do the amplification step?
So I took the extracts that were generated from the first step, and I took portions of those samples and added chemicals to the tubes with these samples, and then I put them onto an instrument called a thermocycler, and this will have the samples go through periods of heating and cooling, and this is to make millions of copies of the specific DNA that we are looking for to be able to visualize it in further down in our processing.
And which equipment did you use for that?
A thermocycler.
Okay. And then how do you use that equipment?
The samples just go on, they are in small tubes, and they will go on into small holes in the thermocycler, and then it is a metal plate that they are in, and it will go through different steps of heating and cooling.
And what happens next to the DNA process?
So the next step will be that those tubes that just went through the heating and cooling, that those tubes will be used for the 4th step of separation.
And who did that step?
Troy Adams.
So you did not do that step?
No, I did not.
And did that end your work on this case?
Yes, it did.
Any cross-examination? The witness may step down. Thank you.
Thank you.
Can you state your name for the court, spelling your first and last name?
My name is Troy Adams, Troyadams.
Thank you. And what is your educational background, sir?
I have a bachelor's degree from Pennsylvania State University in forensic science.
And what is your current occupation?
So I currently work as an investigator for the Lehigh County Coroner's Office in Pennsylvania.
And how long have you been in Pennsylvania?
I've been in Pennsylvania for approximately 3 years now, 2 years working in my current job.
So prior to moving to Pennsylvania, did you work in a different field?
No.
Did you work for forensic sciences in Massachusetts?
Yes.
And can you detail your working history as it pertains to forensic sciences?
Yes. So out of college, I joined the Massachusetts State Police crime lab there as a DNA analyst. I completed all my training protocols and was tested for competency, and then I began performing casework on samples.
And then did you work for a different company in Pennsylvania as well doing forensic sciences?
Yes. After I left the Massachusetts State Police, I joined Quest Diagnostics as a forensic scientist in their toxicology division.
And just keep your voice up so everyone can hear you.
And have you had training for your position at the Massachusetts State Police Crime Lab and also for your position at Quest?
Yes.
And have you had ongoing training as well?
Yes.
And have you taken proficiency tests?
Yes.
And what were the results of your proficiency tests?
I passed every proficiency test.
And do you have any certifications in your current job?
Yes.
And where are those from?
So I am certified as a national medical legal death investigator.
And how did you receive that?
So I received that after a certain amount of training at my current job, and then going through and taking a certification test.
I want to direct you now to when you were working in Massachusetts. Did you do work on lab number 23-00596?
May I refer to my file?
You may.
Yes.
And did you do work on items 9-27.2.1, a sample from interior of a dark gray slipper, 9-27.3.1, stain A on the exterior left gray slipper, 9-32.1.1, the sample from the interior sleeve cup of a Tyvek suit, 9-32.2.1, the sample from stain A on the exterior left pant leg of Tyvek suit, 9-32.5.1, the sample from stain D on the interior right sleeve of the Tyvek suit, and 16-13.1, the unknown tissue recovered from the Swampscott dumpster.
Yes, I did.
And what work did you do on those 6 samples?
So I performed the second and fourth step of the DNA analysis process, quantification and detection.
And can you tell us how you did the quantification step?
So I took a sample, I was given samples from the extraction step, and then I added a mix of chemicals and went through repeated processes of heating and cooling, which quantified the DNA.
And then the process was then next ready for the third step, is that correct?
Yes, it was ready for amplification.
And who did the amplification step?
I believe that was Madison Frank.
Okay. And after Madison Frank did the amplification step, what did you next do?
I took the products of the amplification step and performed detection.
And did you do the detection on those same 6 items that we just discussed?
Yes.
Okay. And how did you do the fourth step, that detection?
I added a mix of chemicals and placed the samples on the detection analyzer.
And then what happened?
And then electropherograms were generated, which were then interpreted by the interpreting analyst.
And where did that data go?
That data was transferred to one of our servers at the State Police Crime Lab.
And did you do the interpreting or did someone else do it?
Someone else performed the interpretation.
Did you do any other work on this case?
Not that I'm aware of, no.
Okay. I have no further questions.
Any cross-examination of this witness? The witness may step down. Thank you, sir.
Thank you.